Hello. Welcome and thank you for joining us today for the BGE-105 Apelin Receptor Agonist for the Prevention of Muscle Aging webinar presented in partnership with Biowage Labs. I'm Jennifer Blinkle, Director of New Product Development and Strategic Initiatives for APS. I'm excited to be here today to share this program with you. Before we get started, there are a few details we'd like to address. First, this webinar is being recorded and a link to the recording will be shared with attendees following the event. To maintain the integrity and respect for your colleagues, we ask that you please refrain from video or photo capture of any part of today's presentation. Please use the Q&A feature found at the bottom of your Zoom screen to submit questions for the presenters at any time. The BioAge team will respond to questions as possible during the presentation and at the end of the webinar. Here are the BioAge presenters that you'll be hearing from today. Now it is my pleasure to hand the program over to Eric Morgan, co-founder and COO, to kick off the webinar. Eric, it's all yours.
Thanks very much for the introduction. So it's a pleasure for all of us to be here speaking to this audience today. So BioAge is a platform-driven clinical stage biotechnology company, and we're advancing a growing portfolio of therapies that treat severe diseases by targeting fundamental mechanisms of aging. We have a powerful human-first discovery platform with key competitive moats, and we have a growing clinical stage portfolio with 3 programs at the moment, and this year is an important year with 2 clinical trial readouts. Next slide. So this is why we work on aging. So if you look at the graph on the right, just taking as 2 examples, the 2 biggest causes of death in most developed countries, cancer and cardiovascular disease. These are very distinct diseases, distinct pathogenesis, symptoms, onset, progression, etc. And yet they share the same underlying risk factor, which is how old you are. And this is the promise of aging science and using aging biology as a source of targets, is that it will give us brand new targets with the potential to affect multiple important chronic diseases. Next slide. And for people not familiar with the space of aging research, it's important to do a little bit of orientation. So this is a relatively new field of study. It was beginning in the 90s that invertebrate research in aging began to really expand. And it was discovered that individual genetic mutations could powerfully modulate the lifespan of C. elegans, Drosophila. And indeed, there are a large number of such examples of genes that modulate lifespan. In the 2000s, we began to see reproducible experiments in mammals, predominantly mice, showing that particular interventions could modulate lifespan and health span, good examples being rapamycin, senescent cell ablation, parabiosis. It's only been in the last 10 years or so that we've been able to study the mechanisms of aging in humans. And because human aging takes place on the scale of decades, we believe strongly that you have to study human aging by acquiring molecular data also on the scale of decades. Next slide. And this is really important to remember that in the population around us, there are examples of people who really have exceptional both longevity and health span. So about 1 in 1,000 people will live to the age of 100. And there's some great examples here. Lila Denmark practiced as a physician to the age of 103, giving her a professional working span of approximately 80 years, which is about twice as much as many of us. And so there's really a lot to learn within the variation that we see in the normal human population between people who age, relatively poorly and these really exceptional lifespan and health span examples. Next slide. And this is a high-level overview of the BioAge platform, which is really rooted in human data and this idea of exploring the mechanisms that separate people who age slowly and people who age more quickly. And so it begins with longitudinal data collected over decades. And we do this, we source the right human cohorts by partnering with really special biobanks that began collecting samples in healthy middle age, decades ago, up to 50 years ago. And they combined this with health records collected up to the present day. So we know everything about the lifespan and health span of these people, how long they lived, what diseases they got, and health span outcomes like physical function and cognitive function. And what we do is we go into these carefully preserved samples and we apply modern omics technologies, proteomics, transcriptomics, metabolomics, et cetera, to really deeply characterize the biology in each of these samples. And this allows us, we're at number 3 here, MAP, to apply our data-first AI-enabled data analysis pipelines to map the key drivers of human aging. When we find something that has a really promising signal in the human data, we move over to our animal platform. So we've put substantial time and effort into creating naturally aged animal models where we can, interventionally, experimentally validate these associations we see in the human data. And for molecules and for pathways that are really compelling, in both the human and the animal data, we take a rapid path to clinic by in-licensing clinical stage molecules that target the pathways of interest that came out of the platform. And of course, we strongly believe that these pathways and molecules will be very relevant to aging in a chronic context. We take them first into clinical trials for severe acute diseases with well-established and acknowledged unmet medical need. Next slide. And I'll just skip over this number slide this time. Great, and this is just the example of Apelin and how it's the subject of today's webinar and how that came out of our platform. Some of the initial key pieces of data that got us interested in this in the first place. So we were very interested in finding targets for muscle aging, and there's a number of outcomes and variables that are relevant to this in our cohort. For example, lifespan, grip strength, walking speed, mobility scores. And so, we took all the variables that we thought were most relevant to this and created a composite predictor. And if you do a histogram of the scores relative to this predictor for all the proteins that we're measuring, you get that histogram on the lower left. And you can see that Apelin is located in the highly relevant tail of this distribution. And actually, it was the only target for which there was a high quality molecule available. And if you dive into the associations for Apelin, you get this graph on the lower right, where we see this very gratifying dose-response relationship between Apelin protein levels on the x-axis and on the y-axis, the chance of increased longevity in blue or physical function in purple. Next slide. Zooming out to the network view here, we can also evaluate Apelin in the context of its correlated molecular mechanisms and the broader pathway. And we see this, again, very confirmatory set level relationship on the right between the set level activation of this pathway and longevity, i.e. all-cause mortality. And so it was this sort of package of human data that got us really interested in Apelin. So we reached out to Amgen, who had a molecule that targeted the Apelin pathway. And we put this into a number of internal mouse models of aging. And we'll be speaking in greater detail later on in the presentation about this. So I'll just for now, I'll give you a very high-level summary of just one of the experiments. So this is a casting experiment in mice where mice are casted, they get muscle atrophy as a result. And this molecule was able to essentially completely prevent the atrophy associated with the intervention. Next slide. And so as a result of the totality of the interventional animal data package and the human data, we decided to in-license BGE-105 from Amgen for development in muscle aging indications. And indeed, we have initiated a phase 1b trial in healthy elderly where we will be putting volunteers at bed rest, and we will be measuring key muscle atrophy and function-related outcomes, as well as proteomics. Next slide. And just to zoom out to see, how this fits into our pipeline, we have at the moment 2 programs in muscle aging, 2 in immune aging, and the ones with clinical readouts this year will be BGE-105, which we're developing for muscle atrophy, and BGE-175, which we are developing for viral pneumonia. Next slide. And just to give you a few more details about this molecule and program. So this really has the potential to be a first-in-class molecule for muscle aging with a novel mechanism. The molecule is an APJ receptor agonist. It's an oral small molecule with once-a-day dosing, clean safety profile already previously in phase 1. And at the bottom here with a package of preclinical data that not only supports acute muscle atrophy type indications, but also chronic muscle function and sarcopenia related indications. And as a result, you can see on the right that our planned indications begin with ICU-associated diaphragmatic atrophy, but we plan to expand to other muscle atrophy indications and ultimately to sarcopenia and frailty. And as I said, we expect initial clinical data by the end of the year. Next slide. And now I will hand this off to Patrick Martin.
Thank you, Eric, and thank you for the opportunity to speak with you all today. I'd like to give you a little more background on the BGE-105 molecule and the in vitro data demonstrating that it is a potent and selective agonist of the APJ receptor. Apelin is a peptide hormone first identified in 1998. It's encoded by the Apelin gene and is a GPCR agonist. In humans, it is one of the 2 endogenous ligands for the G protein coupled APJ receptor that is widely expressed at the cell surface of various organs, such as heart, lung, kidney, liver, adipose tissue, in the GI tract, the brain, the adrenal glands, endothelium, and in human plasma. We see from these data that the APJ small molecule agonists, AM8123, also originally an Amgen molecule, and BGE-105 activate the APJ receptor at similar or lower molar concentrations and in a similar manner to the endogenous peptide ligand Apelin 13. This has been demonstrated by a number of APJ-specific GPCR assays. We see increased intracellular CAMP levels induced by APJ activation, human GTP gamma S binding assay that measured G-alpha protein stimulated receptor activation, APJ-mediated beta-arrestin recruitment, which is the translocation of beta-arrestin from cytoplasm to plasma membrane, and finally by APJ receptor internalization following receptor activation. This is the APJ beta-arrestin complexes formed, then internalized into cytosol, form intracellular vesicles happening within 30 minutes of activation. So together, these give a very, very strong picture of this novel small molecule behaving similarly to the endogenous apelin peptide ligand. To further document the beta-arrestin recruitment and beta-arrestin receptor internalization findings, these were also reproduced using several different assay models. Additional assays for other receptors such as AT1, beta-2 adrenergic, and other receptors were conducted to confirm the selectivity for APJ. So in summary, what we see is that BGE-105 is behaving as a full apelin agonist that activates the APJ receptor with same or greater potency as the endogenous peptide ligand, and is capable of triggering the beta arrestin recruitment and the receptor internalization. I'd now like to introduce Dr. Bob Hughes, VP of Biochemistry at Biowage, who will walk us through more of the non-clinical data for BGE-105.
Thanks, Patrick. Let's go to the first slide. So Patrick gave a nice introduction to apelin APJ biology. So we're going to zero in a little bit more on its role in muscle function. So what's shown here in this cartoon is in the left panel, you have a muscle, a piece of skeletal muscle, and it's expressing the APJ receptor, which is responsive to endogenous apelin. And the endogenous apelin could be originating from any number of tissues. It's very widely expressed, but it's also an autocrine function where the muscle contraction is actually known to produce apelin in the skeletal muscle, and it will, through an autocrine-like mechanism, impinge back on the muscle to promote its growth and contractility and energy metabolism. And this is the reason that is often referred to as an exerkine, which is essentially a hormone that's produced by muscle contraction and has effects on muscle physiology. So in the middle panel, you can see in the young muscle where apelin is abundant and potentially activity is higher, the skeletal muscle is experiencing a high level of signaling through APJ, and that activates a number of pathways, but most prominently AMPK, which is involved in a number of things, including energy metabolism, autophagy, and mitogenesis, AKT, which is kind of broadly impacting a lot of pathways involved in protein synthesis, as well as cell survival through the mTOR pathway. And then PAX7, which is expressed in stem cells, including muscle stem cells. And muscle stem cells actually also contain APJ. Now, I'm going to get to Cedric Dre, but he's the one who discovered, Cedric Dre, our collaborator, the role of, sorry, I didn't mean to go ahead. Can we go back one? Sorry, I was just foreshadowing there. So we can see that in the young muscle, we have lots of apelin signaling, lots of APJ downstream signal transduction as compared to aged. And what others have shown is that the apelin peptide, if supplemented, can essentially rejuvenate aged muscle. And we've been able to show that BGE-105, which is the orally available small molecule, essentially functions as an apelin mimetic in its role to rejuvenate aged muscle. Next slide, please. Okay, so a lot of the work that we've done over the past few years has been in collaboration with Dr. Cedric Dre, who's in Toulouse. And as Patrick alluded to, the peptide was originally identified in 1998 at Takeda Pharma in Japan. And it was really just a ligand for an orphan receptor. It was then appreciated to have multiple roles in various tissues, but it was Cedric and his colleagues in Toulouse in 2008 that really demonstrated that it had a very powerful impact on muscle and on aging and muscle contractility and a number of other factors, including muscle regeneration after injury and stem cell mobilization. So there's a wealth of data around this, but we've just chosen to present this one aspect of of Cedric's paper from 2018. Sorry, I said 2008. And this showed a lot of pleiotropic beneficial effects in muscle and aging in mice. And what's shown here is treadmill maximum speed activity of young, middle-aged, and old mice, 3, 12, and 24 months. And you can see that there is a decline in the maximum speed, particularly in the 24-month-old mice. But that can be rescued by the application of apelin peptide and restore the maximum treadmill speed back to a more youthful speed. And in the right pair of the graph, you can see that in the apelin knockout, even in a young animal, you see a significantly reduced mobility that can be rescued, again, by putting the apelin peptide, supplementing the apelin peptide. And so this is just one example of how apelin seems to be affecting muscle physiology and muscle function, in this case measured by mobility. And we'll be presenting some mobility data later. Okay, next slide, please. Okay, so Eric Morgan foreshadowed this particular experiment, but I think it's an important one, that BGE-105 prevents muscular atrophy during immobilization. So in this experiment, you take a mouse. The way this was done was we used 20-month-old mice, which are reasonably old, and casted them for 3 weeks on one limb. And essentially what you can see in the middle panel is that after the 3 weeks of casting, you can see a significant decrease in the total weight of the tibialis anterior muscle and in the casted limb versus the non-casted limb. But now, when the animals have BGE-105 administered orally during that 3-week casting period, it essentially completely abolishes the atrophy. And these data were done in collaboration with Ramsey Kairala at Myologica, which is a CRO in Maryland that specializes in muscle physiology. So I think this is a very important result because it's a model of muscle atrophy and immobilization, which occurs in hospitalized patients, bed rest, et cetera. So I think it's a very powerful predictor, if you will, of success in immobilization, atrophy, amelioration. Next slide, please. Okay, so one of the experiments that was reported by Cedric Dray in his 2018 paper was that apelin peptide could rescue or accelerate recovery from cardiotoxin injury. And this experiment is cartooned in the left. You take a mouse, you inject it with a toxin that leads to muscle necrosis, and you simply measure the amount of time and the molecular features associated with the recovery. And so we've looked at a lot of different parameters here, but I'm just showing you a subset of those. So in the middle top panel, you can see the cross-sectional area of the TA. That's the cross-section of the muscle fibers, which is an indication of repair. And you can see that there are 2 sets of data, 3 days post-injection and 7 days post-injection. And the cross-sectional area with PBS, which is the vehicle. Apelin, you can see, has a pronounced and significant effect both at 3 days and 7 days in enhancing that anatomic measure of repair. And BGE-105, again, administered orally is arguably, is equivalent to and arguably better than the apelin peptide, which is administered intravenously. And I think that's a really important point about 105, is that it's an oral mimetic of the peptide. And some of those data, the histologic correlates, are shown in the upper right. You can see the effect of what's happening as compared to control in apelin, and 2 different doses of 105. And what you generally can see here is an enhanced H&E staining, and also a more organized muscle cross-section in terms of fibers. Now, in the lower 3 panels, in these in this experiment, we measured molecular markers of regeneration, PAX7, which is expressed in quiescent stem cells and is associated with their mobilization into a differentiation program. PAX7 actually interacts with MyoD, which restricts the differentiation program into a muscle sulfate, and MyoG is another muscle indicator. And what you can see in all 3 cases is that in the 7-day, in the case of PAX7, after 7 days, you can see a greatly enhanced expression of PAX7, which indicates that quiescent stem cells are becoming committed to differentiating. And the MyoD result shows that the PAX7 effect is exerting a commitment to muscle cell differentiation. So these data taken together indicate not only that 105 can accelerate repair from injury, but that repair is dependent upon mobilization of quiescent stem cells or muscle cell precursors. Next slide, please. Okay, so that was all from mice, but now we wanted to, kind of verify some of this in vitro. Now, it's difficult to do similar experiments in humans, so we went to an in vitro model, which are essentially human myoblasts that are cultured in vitro, that are obtained from biopsies from old donors. This experiment was done by Cedric Drey in Toulouse, where they have a hospital setting where they can obtain these muscle biopsies. In this case, so what's shown in the upper left cartoon is the progression of satellite cells, which are essentially the stem cells, into myoblasts, myocytes. They organize into myotubes and then form the myofibers. And below that, you can see the expression of the kinetics of the expression of the various factors, transcription factors that are associated with the different stages of differentiation. So in the last slide, I showed you the PAX7, which is in the satellite cells. And then by the time you get MyoD expression, you're already committed to becoming a myocyte and on your way to becoming a myotube. So we did this experiment with Cedric, where we took these human myoblasts and in vitro and culture, and they can be induced to differentiate into myofibers by certain culture conditions. And we wanted to see whether, and it was known that apelin peptide could accelerate this process, and we wanted to see whether 105 could, and that data is shown below. We have images, but I think this molecular data tells the story. In the lower right, excuse me, the lower left, you can see the amount of proliferation that's going on. That's just simply cell count at day 4. And you can see that apelin does enhance proliferation in this experiment, not significantly, but 105 does significantly increase the rate of proliferation. But more importantly, I think the PAC7 and the MyoD data really show that BGE-105 is equivalent to apelin in its ability to promote the mobilization of the satellite cells and their subsequent differentiation into myocytes. So this is completely consistent with what we observed in the mouse cardiotoxin models. So it shows that human cells in vitro can recapitulate the apelin mimetic effects of 105 that we observed in the mouse. And with that, I will turn it over to my colleague, Justin Rebo. He's our Senior VP of Translational Aging Biology.
Hey, everyone. Thanks, Bob. You can go to the next slide. So at BioAge, we think it's very important to test potential age-related disease interventions in naturally aged animals. We use naturally aged animals because they are much more translational for age-related disease compared to more artificially induced models. For many disease-specific assays, we are working closely with CROs or academic partners like Dr. Cedar Trey, who are the experts in those diseases or those specific models. But our own internal core competency is in aging itself. To that end, we have set up a 6,000 animal vivarium full of aging animals. We are set up for a wide variety of short, medium, and long-term assays in these animals. Often for the more medium and long-term assays, we're delivering therapeutic interventions long-term or in the food or the water if we can, to minimize the kind of interactions we have with these animals as much as possible so we can observe their pure phenotypes. And we are able to look at a wide range of age phenotypes. Next slide. Well, actually, can you go to the next slide? I'll come back to this one. We have developed extensive capabilities to measure a wide variety of aging phenotypes. 24-hour automated activity monitoring, strength assessments, a wide variety of behavior assessments, 28-point clinical frailty index, measures of metabolism, immunity, neurosensory function, and these are just a few examples. So now you can go back to the previous slide. So we tested BGE-105 in this system. We really use this system as a medium throughput first pass screening tool. We started with already aged animals at about 2 years of age, and we gave BGE-105 in their drinking water for a 3.5 month period. We saw a significant protection from the decline in spontaneous activity with age in animals treated with BGE-105 compared to control animals. Which we assessed again using an automated system where the animals are monitored 24/7. These animals maintained their muscle mass in the middle panel significantly relative to control animals as they continued to age. And on the right panel, we saw a significant decline in strength as assessed through the grid hang assay relative to control animals. Whereas the BGE-105 treated animals maintained their strength. They didn't change over this treatment window. So we really see a very wide range of highly protective effects of BGE-105 in the muscular system and in other systems as well, and in many preclinical models, including this kind of more broad, really frailty assessment that we use internally, and as well as the work that Bob was kind enough to take us through. And with that, I believe you can go to the next slide. Next slide. With that, yep, I'll turn it over to Dr. Carrie Furr, our SVP and Venture Head of the Apelin Muscle Aging Program. Take it away, Carrie.
Thanks, Justin. Our near-term development plan is to focus on acute muscle atrophy indications, such as diaphragm atrophy associated with mechanical ventilation. More on diaphragm atrophy will come after the next slide. In the longer term, BGE-105 could be developed for chronic aging indications, such as sarcopenia and frailty. We're excited to share that the phase 1b randomized, double-blind, placebo-controlled single and multiple ascending dose study to characterize the pharmacokinetics and pharmacodynamics of BGE-105 in older adults is ongoing, as announced in a press release last week, and as depicted in the far left panel here. Building off of existing clinical data, BioAge's phase 1b clinical trial will be studying up to 72 healthy older adults. Treatment in the multiple ascending dose segment of the study is during a period of bed rest. One of the key hallmarks of metabolic health is insulin sensitivity. Earlier studies have demonstrated that short periods of bed rest impair glucose tolerance and lower insulin sensitivity. One of the objectives of the trial is to assess mechanisms that may contribute to the development of insulin resistance during short-term muscle disuse, and this takes into account the expected muscle atrophy. Study endpoints include safety and pharmacokinetics of different dose strengths, as well as pharmacodynamics, specifically the hemodynamic effects of apelin receptor agonisms and parameters of skeletal muscle atrophy. In an exploratory manner, we will also assess multi-omics profiles. The phase 1b results will inform subsequent clinical development. To demonstrate proof of concept, we plan to test the ability of BGE-105 to prevent diaphragm atrophy that develops during mechanical ventilation due to disuse. The diaphragm of patients undergoing mechanical ventilation atrophies, with 40 to 75% of mechanically ventilated patients developing clinically significant diaphragm atrophy. Diaphragm atrophy typically begins to develop within 24 hours of mechanical ventilation, with most profound changes occurring within 3 days. Diaphragm atrophy is the leading cause of difficulty weaning from mechanical ventilation and is associated with poor clinical outcomes and increased resource utilization, including longer time on mechanical ventilation, longer time in the intensive care unit, and higher mortality. Unfortunately, there's no therapeutic prevention or treatment of diaphragm atrophy, although there are ventilation approaches that aim to minimize lung stress and limit diaphragm injury. To connect back to the illustration of our clinical development plan, the outcome of the phase 2 proof of concept study will inform pivotal study design or phase 3 studies to substantiate or support marketing application to address this unmet medical need and enable subsequent development of BGE-105 to expand the label to include other acute and chronic muscle aging indications, including sarcopenia and frailty. And I'll hand it over now to Dr. Paul Rubin, our Chief Medical Officer, to expand on BioAge's clinical strategy. Paul?
Thank you, Carrie, and thank you all for attending this meeting. Go to the next slide. So obviously, BioAge is very interested in ultimately being able to alter the pathologic course of aging, especially to prevent those things that occur as a result of aging that affect quality of life, health, lifespan. However, it's from a clinical development perspective, it's difficult just to jump right into that aspect of it. So the way that we look at this is for our initial clinical experiments, we're trying to find tractable indications that are related to these chronic pathologies that develop as a function of aging. They're tractable, that they have validated endpoints and allow us to do a controlled trial that's actually manageable and teach us about how to ultimately develop drugs that can prevent the sequelae or the sequelae of aging. So our first foray into the clinical with these clinical trials with this mechanism is to really define these well-controlled, doable trials. But again, aspirationally, we're very interested in altering the pathological aspects of growing older. Next slide. So it's sort of how we, ended up. Sarcopenia obviously is an important component of aging and ultimate morbidity and mortality associated with getting older. So as many of you know, young, healthy muscle has a certain muscle volume, but as you get older, you develop a significant decrease in muscle mass or sarcopenia that ultimately is a major component of frailty. And as you can see on the right, this decrease in muscle mass continues very briskly as a function of aging. You can see that, and it's clearly measurable, and it's also very important. Next slide. Sarcopenia is a major aspect of aging, the loss of muscle mass, which prevents the ability to function in the same capacity as you can when you're younger. So this concept of frailty has become very important and it's starting to be very well studied and starting to be understood better. And there's a number of components to frailty. Obviously, there's weight loss, and part of that weight loss relates to loss of muscle mass. Patients as a function of aging and developing frailty become very easily exhausted with low levels of physical activity. This can actually be measured by walking speed. And in fact, walking speed, as I'll show in a second, does correlate with morbidity and muscle weakness. If you look at the representation on the right, there are multiple causes that ultimately lead to sarcopenia and then multiple sequelae resulting from the sarcopenia, all of which are detrimental. Next slide. And then it's clear that this condition occurs as a function of age. And as you're getting over the age of 80 or 85, there's a very high percentage of people that suffer from this particular condition, thus just underscoring the importance of determining ways that we can modulate this process. And as you can see, women are more frequently affected than men. Next slide. Now, we talked about gait speed, and gait speed is something that is relatively easily measurable. And in fact, this particular graph suggests that it should be part of routine screening as patients get older, because your gait speed clearly affects mortality. It correlates with mortality. So having a metric that people can actually use to determine the state of their underlying physical function and frailty is an important tool that should be incorporated into routine physical exams, especially of the elderly. It also becomes an objective endpoint for any pharmaceutical modulation that any studies that we might put in place. We can use this as a metric for determining success. Next slide. So it's interesting because frailty is kind of a novel concept. And it's really important, ultimately, if we would like to get pharmaceuticals developed and ultimately approved for the treatment of frailty, that the various regulatory agencies around the world have to acknowledge that this truly is a syndrome that warrants drug therapy. And in fact, the EMA, the European regulatory body, has come out very clearly saying that this is an important indication, and they're starting to work with both academic institutions as well as pharmaceutical companies to try to acknowledge that this is important and determine ways that we can go forward to develop important drugs to treat these patients. Next slide. So it's interesting, and as I mentioned before, that it's really difficult to just start with the indication that affects hundreds of millions of people worldwide. And in fact, you really have to start small and then expand as you learn about it, but also as your compound, if you do develop it, you have additional data showing not only that it's effective, but that it's safe in larger groups of populations. And a case study of how to go about doing this is the way the statins were developed for the treatment, ultimately, of cardiovascular disease. But it started with just approval in a very small segment of familial hyperlipidemia, where it showed that you can improve that and improve the various lipid abnormalities seen in blood. But as we had more experience in using statins, we understood how these work, that they are safe, how to dose them, and we were able over time to expand the utility of these particular drugs, where ultimately they're one of the most commonly used medications in the world. And this is ultimately how we need to go about developing these compounds, starting with these small tractable indications and ultimately learning how to treat the broader population, which literally would include hundreds of millions of people worldwide. So one of the ways we're approaching this, and I think Carrie alluded to this, is where apelin has an effect on skeletal muscle. What is a small, highly morbid condition where muscle atrophy is important to the development of significant morbidity and also mortality? So we landed on atrophy of the diaphragm in patients that are undergoing mechanical ventilation. Patients that are intubated, and this is disproportionately affects elderly. When they are intubated, they undergo profound atrophy of the diaphragm, and it occurs literally within days. So we believe that this is a clinical model that not only is important in terms of the number of people that ultimately suffer from this, but could teach us how to use this drug to prevent atrophy of the muscle. Because again, in these ICU ventilated patients, not only can we look at the diaphragm, and in fact, you can measure diaphragmatic size using ultrasound, but these patients also are at profoundly strict bed rest, so we can look at peripheral atrophy as well. And if we can understand that, that could definitely teach us how to treat frailty. So that is the pathway that we want to follow for this drug. Now, you also have to have indications that are acceptable, have acceptable morbidity, and have clearly validated and tractable endpoints. So, for example, just using patients with bed rest, where you might show an effect in the absence of mechanical ventilation, you might see effects on the muscle atrophy, but it would be very difficult for that alone to move the needle in terms of a regulatorily approvable endpoint. So that's why we go to a more severe group at this point in time. Next slide. So in summary, I mean, at BioAge, as you've heard from my colleagues, we are trying to map human aging, especially relates to various things that you can measure within blood and tissue. We have very unique data sets that allow us to look at patients premorbidly and to determine what factors within their bodies can ultimately predict what will occur as a function of aging, and then identify those targets and hopefully find drugs that can modulate these particular targets. And this is how Apelin emerged. We've done extensive work on our Apelin mimetic BGE-105, and we know that this is a very potent drug. It can be delivered both orally as well as intravenously. We have amassed quite a bit of preclinical data that kind of validate the concept that the use of an Apelin mimetic can affect muscle mass and muscle function. We are going to start with, we think, a very relevant disease that has a regulatorily acceptable endpoints, ultimately looking at clinical recovery from mechanical ventilation. And using that data, our hope is we'll understand how to treat the broader patient group that ultimately suffers from sarcopenia and frailty. And I think that's where we'll end right now. We have quite a bit of questions that have been brought in, and I'll try to triage these questions to the various group. And I want to thank you all very much for your time. And I don't think we can get to all the questions, but I'll try to get to many of them. Okay. So we'll start with, let's see, I have a list here. This is probably for some of the preclinical scientists. The question is, other than exercise, what regulates apelin synthesis and release? And in this regard, does exercise training mimic apelin receptor agonism? And I think maybe Bob, Justin, or Patrick can take that question.
I'll address that. So, as Patrick mentioned, apelin is very widely expressed expressed throughout the body in adipocytes, in the brain, in the liver, but also in the muscle. So we haven't really investigated the idea of, what are the other activities or physiologic parameters that could enhance apelin expression in other tissues. We become very focused on Cedric's initial observation that muscle contraction will increase apelin expression in the skeletal muscle itself, and presumably has at least an autocrine effect and probably has an exocrine effect on distant tissues. I think for what they're widely reported observations that resistance exercise can improve cognition. So there may be something going on there in terms of endocrine, effects of this exerkine. But we're primarily interested in the autocrine effects on the muscle itself by apelin that's expressed as a result of muscle contraction. I don't know if that answers the question, but.
Okay, thanks, Bob. Here's a simple one. Probably Justin and Bob can answer these. Are these experiments only using male mice?
Here I can answer that. Sorry, there we go. No, actually, for our longer term experiments that we did in-house, those are all female mice. Some of the experiments, and Bob can chime in with more detail here, that we did in collaboration with Cedric Trey, and I believe we're in male mice. But we certainly see a good phenotypic change in both male and female mice.
Yeah, the data that I presented that were done in the Dre lab were with male mice, old mice or aged mice, and as Justin said, our frailty assay done in-house was done in females. So I don't think there's any evidence either from our experiments or from the literature that there's any big difference between male, significant difference between response to apelin or BGE-105 in males versus females. In fact, our data suggests that there's not.
Okay. Another preclinical question. Apelin receptor links to GI and can interact with beta-arrestin. How does the expression of apelin receptor compare with that of other GI-coupled Gpcrs in skeletal muscle?
That's an easy one. I don't know. We haven't looked, but it's an interesting question.
In your CTX injury model, did you look past 7 DPI, 30 DPI, for example, and did you measure muscle function or contractility at this time?
No, we didn't look past 7 days. And in those injury models, we just purely looked at molecular markers and histologic markers of muscle repair. We did not do any functional assays in the CTX experiments.
Were any
also,
go ahead.
Sorry. No, I think also you can see from the histology that Bob shared that at 7 days in that particular model, the healing process was mostly complete.
Okay. And in a related question, were any ex vivo contractile function experiments performed in the Murian model?
No. But, as long as we're talking about CTX, I think it's an interesting fact that the acceleration of repair after the CTX injury only had an effect in the old mice, and it didn't have any effect in the young mice. The young mice apparently have an endogenous kind of maximal repair kinetics, and what the BGE-105 does and the apelin is bring the repair kinetics back to a more youthful state. I noticed in the questions asking about whether the 105 could cause hypertrophy, muscle hypertrophy in the younger animals. It's a very interesting question. But in the CTX model, there was no effect in the young mice. So it just restored youthfulness or a youthful phenotype in the old mice.
Any effects of apelin in non-skeletal muscle? And does apelin prevent apoptosis of skeletal muscle? And this is related to your answer, does it stimulate growth and regeneration?
Well, we know that it stimulates growth and regeneration in the CTX model. We did not look at apoptotic markers. It's an interesting question whether it's anti-apoptotic. I don't believe I'm not aware of literature suggesting that apelin is anti-apoptotic. I think that the CTX is mostly a necrotic form of tissue death, but it would potentially be interesting to look at apoptotic markers, but we didn't do that.
Okay.
But yeah, I mean, beyond skeletal muscle, maybe Patrick would like to say a word, but it has effects in cardiac muscle, the pathway.
Right.
No,
Patrick,
I don't think I had anything too much to add to that, Justin.
Does BGE-105 affect the activity of myostatin?
We didn't look at it, and I'm not aware of any literature that reports an effect, but it is an interesting question.
Okay. Does your apelin mimetic induce more myotube fusion in your in vitro assays? I think you're on mute, Bob.
Oh, sorry. Yes, it does, but we didn't show that data.
Okay. Justin, did you measure and separate nighttime versus daytime spontaneous running?
Absolutely. We monitored activity, 24 hours. And I can tell you this, almost all the activity for a mouse is in the night. They're primarily nocturnal animals. One thing that you can notice with aged animals is that their activity will start to bleed a little bit more into the light cycle rather than more exclusively at night with younger animals.
This question, did you check which cells and muscle drive the in vivo response to BGE-105? This questioner says they have data in press suggesting that endothelial cells and muscle are highest APJ expressors and are very important for apelin effects.
That's a great question. We did not look at increased any increased vascularization or vascular function. I mean, it is known that, and maybe Patrick could comment on this, that in humans, BGE-105 does improve hemodynamic parameters. The data that we have is what was presented is that we do know for sure that it impacts the stem cell population, but given what's known in the literature about APJ expression in endothelial cells, it's quite likely that if we were to look at endothelial cell function during muscle repair, say in the CTX model or even in the frailty model, that there's a reasonable expectation that we would see improvements in endothelial cell related functions.
Thanks, Bob. There's a number of people who have asked if this drug is orally bioavailable or is it just parenteral? And in fact, there are both orally bioavailable formulations as well as IV. And it's been looked at both, and there's very good oral bioavailability of the compound.
Our own studies were delivered, our long-term studies were delivered in drinking water. So it's very orally bioavailable.
Yes. Okay. Let's see. Those are the ones that there's been a number of questions that have come up lately. There's a question related to our trials plan with respect to heart failure. Patrick, you want to take that one?
Sure. So Amgen who was the original discoverer of this molecule, that was actually their plan for their clinical program. And they looked at some heart failure patients in their phase 1 program. And it was a fairly short term and a small number, but they did not see the magnitude of effect that they had determined would be necessary in order to continue development. As part of our ongoing phase 1 work at the moment, we're continuing to look more fully at the various hemodynamic effects of BGE-105, but at the moment, we are not planning further heart failure work.
Thank you. Okay. So question just for the field. Are there any substantial differences regarding the immune response to muscle injury between treated versus untreated, or differences in general inflammation with age with treatment? No. I think
we didn't report that, but there are indications, unpublished indications, that apelin peptide is anti-inflammatory, and there's no reason to expect that those anti-inflammatory effects of apelin peptide, that BGE-105 would not also have those effects.
Yeah, Bob, there were a few questions related to the fact that, especially in elderly or in certain situations, chronic inflammation might affect muscle atrophy. And certainly we believe that sarcopenia and frailty, there might be a chronic inflammatory component of that as well. And it's something that we at BioAge are interested in studying, the role of inflammation and muscle atrophy and as well as frailty. So
I don't know that there are any. I'm not aware of studies that specifically address the question of whether apelin somehow dampens the negative effects of chronic inflammation on muscle. But it would be surprising if that were not the case, that chronic inflammation was a significant factor in sarcopenia. I mean, if it were not, I would be surprised.
Yeah, but I think it's an important question, and one that obviously we believe this may deal with, but we'll continue to look at closely. Here's a question probably for Patrick. Do you know what the exposure or concentration of 105 is used in our in vivo studies, and is it higher than the physiologic concentrations of apelin? And did we observe side effects?
So we have studied doses now that achieve concentrations above what had been the EC50s and EC90s from the non-clinical data. So we believe that we have achieved concentrations that should be giving us significant receptor agonism. So far in the clinical program, there have been almost no adverse events or safety findings. It's been very, very well tolerated, both the oral and the IV. And we have not seen any serious adverse effects associated with it. So obviously, this is something that we will be looking at carefully throughout the entire development. But we've achieved, I believe, substantial exposures. And I guess the one other component of the question was, is this higher than the endogenous levels? You can't really compare directly on a concentration basis, but in terms of the amount of agonism, yes, we believe that we would have reached sort of full agonist doses.
And I could also add that in vitro, looking at GPCR or APJ activation, the EC50s in terms of they're comparable to Apelin on a molar basis, and what we chose doses for our preclinical studies that would result in full agonism, maybe a little bit above that. And I'd also add that the preclinical tox data that was done at Amgen, they were not able to see or observe any toxicity in preclinical animal models, even at very high doses of 105. And in fact, the very experienced toxicologist at Amgen said that this was one of the safest drugs that he's ever worked with in his career at Amgen. So that gives us confidence that the toxicity may not be a big problem with this particular drug.
Yeah, we have a lot of flexibility in our clinical trials as to exposure, and we intend to obviously push the dose until we see a plateauing of effect, which I think is an important aspect of our phase 1 trials as we go forward. Bob, here's a question that I know you and Cedric Ray have thought about in the past. Were histologic studies performed on the bone of the laboratory animals? It is known that sarcopenia is highly associated with the development of osteoporosis.
Yes, that is something that we're very interested in, and I will say this: that there's a published paper looking at the effect of apelin peptide on bone repair in mice in a bone injury model, and it's pretty clear from that publication that apelin peptide will accelerate the healing of bone in a particular model where they drill a hole into the tibia and then measure the kinetics of that hole being essentially repaired. This was a study done by some orthopedic surgeons. There's also unpublished data that not ours, but that I'm aware of, that apelin peptide does promote bone healing. And so that's a question that we're extremely interested in. And our expectation, once again, is that if apelin peptide promotes bone healing, there's a reasonable expectation that 105 will too. Also, apelin peptide in vitro promotes or accelerates the differentiation of osteoblasts into calcified bone cells. So there's a strong expectation that there's going to be a positive effect on bone healing and bone health.
Thanks, Bob. There's a question regarding the fact that AP13 has a short half-life, and how does that compare to BGE-105? And we have actually kinetics in humans, and that's one of the beauties of this particular compound, is that it has an elimination half-life of about 19 hours. So it's very stable and should provide, long-term exposure. So it'll be interesting to see what happens as a result of that. Here's a question: is apelin, is it conserved across species, mice, dogs, human, etc.?
Yes, it's highly conserved, and I think the active, what we believe to be the active form, which is the 13, or sometimes referred to as apelin 13, which is processed out of the precursor, which is 70 amino acids. I think that it's certainly highly conserved. It may be 100% identical between, say, dogs, rats, and mice, but I'm not certain. But it's very highly conserved.
Okay. See if there's any other questions. Yeah, one question is intramuscular delivery an option as well? We haven't looked at it specifically, but it probably can be delivered intramuscularly, but we haven't looked at bioavailability through that mode. Question, in the Bedrest study, how will you measure insulin sensitivity? Patrick, do you want to answer that?
So right now, we are looking at fasting glucose levels. We are looking at insulin levels as well as calculation of HOMA-IRs. There are, this study is not designed specifically as a metabolic study in this regards. So we're hopeful that we will at least be able to see trends there. Clearly, if we were to do a specific study where we wanted to assess this more thoroughly, we would be much more rigorous about diet and things such as that, as well as stabilizing a baseline. But in the current study, we are basically running people in for a couple of days before we start measurements, and are hopeful that that we will be able to get data that will be able to demonstrate at least a trend towards an effect after 5 days of dosing.
Yeah, just to follow up, we are in our Bedrest study, we are trying to have only patients over the age of 65. So hopefully they'll have a higher degree of insulin resistance at baseline, which will allow us, I think, more room to see an effect of the drug on insulin sensitivity as well, I think, in looking at it. Here's another question. Do you think BGE-105 will work differently in slow twitch fibers versus fast twitch fibers? Any thoughts?
If you look at, we didn't present this data, but there are different magnitudes of effects in different skeletal muscle types, and those effects are not obviously attributable to those differential effects are not obviously attributable to fiber types in those different muscle subtypes. But it is an important question, because I think degeneration or atrophy is known to be associated with greater or lesser atrophy is known to be associated with fiber types. But we don't have any evidence that at least 105 is differentially impacting different fiber types.
Thanks, Bob. This is one last quick question: is how many days will patients be in bed in the phase 1 trial? And I believe that's 6 days, which is about what we can do practically in normal volunteers in a phase 1 unit. Otherwise, it would be very difficult to get people to comply. I think we're going to end on that note. So as you can see, here is the information related to contacting BioAge, either for potential collaborations, and we're very interested in people's ideas as pertains to further understanding the role of an apelin mimetic in the treatment of disease. And we are a growing biotech company, and we are in the process of looking for qualified individuals to help us with our mission. So you also see the contact information for our senior vice president of people. Thank you all very, very much for your kind and rapt attention, and the very thought-provoking questions that has given us a lot to think about as well. And I hope you have a great rest of your day.
All right. Thank you, Paul, for organizing and facilitating all of the question answering. That was a great group of questions, and I am impressed with how many you all got through. Thank you to the BioAge team for sharing your work with us today. That was really exciting. It was great to see the engagement and the involvement. And thank you to the participants for joining us for today's program. We ask that you please complete the webinar survey to provide feedback on today's program and inform APS's future programming. As a reminder, we will email a link to the webinar recording when it becomes available. And thank you again for joining us today. We hope you have a great rest of your day.
Thanks, everyone.
Thank you, everybody.